So in 18.1, we talk about how bacterial cells regulate their gene expression through the operons. Now, human cells or eukaryotic cells don't really do the couple of similar examples, but for the most part, we don't have the operons in cells. Instead, we have a bunch of different stages at which we regulate what genes we're going to make. So, as you know, we start off with DNA, we transcribe it into RNA, gets a little bit processed, so then it goes through translation, and then we have proteins. So, at each of these different steps, there's things that we can do to regulate what protein is made and the job that it does. So, okay, so the reason for this, like what does that mean, why does it matter, is because we have all of our cells have all of our chromosomes in them, right? All cells have all 46 chromosomes except for your gametes, which have 23. But they have all of them in them, but they're obviously not all making the same genes because your eye cells are certainly not doing the same thing as your tongue cells or as your heart cells or as your liver cells or your skin cells, etc., etc. So they have very specialized jobs, and that's based on whatever proteins got turned on. So our eyes have the kinds of proteins in them that form the lens that are connected to your optic nerve that make a picture in your brain. Your heart is made up of muscle that pumps blood. So they have very different jobs, and that's because of gene regulation. So in our eyes, we turn on the genes that make them our eyes, and in our heart, we turn off the eye genes and on the heart genes. And so that's why. So in any given human cell, it's mostly about 20% of genes are turned on or expressed, to use the technical term. But our specialized cells, like our brain cells or our heart cells, are even less. Like very few of all the possible genes are turned on because we don't need the other ones. We only want the ones that are going to do the job that is intended for that cell. So, like I said, almost all cells in an organism are genetically identical. And the differences in these cells is due to what we call differential gene expression, which means that in each cell, we're turning on different genes, like I just explained. And so all our cells have the same genome, which is all 46 chromosomes in the same cells that we have. But they all do different jobs, and they all have different genes turned on. Any errors in this process, so based on like regulating gene expression, can lead to cancer. And we'll talk about that later, but or any other disease, or a number of other diseases. But gene expression is regulated at many stages, like I just said. So here's the whole big picture of regulation of gene expression. So we're going to start at the very beginning with how we package our DNA, how available it is. Then we're going to go through transcription, some RNA processing, how it gets to the cytoplasm, the translation of the RNA, whether or not the RNA hangs out or not, or if it gets degraded, then it being turned to a protein and what happens to it as a protein, how long it's a protein, etc., etc. So I think, okay, so the basic, no, we'll come back to this. So the first one is regulation of chromatin structure. So remember that DNA is packaged into what's called chromatin, which is a complex of DNA and proteins that help kind of organize the DNA. Because think of it this way, if you tried to fit an entire ball of yarn into a tiny little Ziploc bag and it wasn't organized, you just shoved it in there, and I said, I need that specific piece of yarn right there, and you pulled it out, it'd be all tangled. So we organize it very, very well with proteins. And the proteins that we use are called histones. So histones are what organize the DNA. And so we can either pack the DNA super, super, super tight together to save space, and kind of like how you don't need all of your parents' old yearbooks from high school out on the shelves. You can put them in a box and put them in the attic. It's still there. The information is still in your house, but you don't need it right now, so why have it out? You're going to put as many yearbooks in one box as you can, put it upstairs, or put it in a closet. And so that, when it happens to chromatin, is called heterochromatin. It's very, very, very tightly packed, and the genes that are in there are not easy to get to. So they're not usually expressed. So that would be the cells, that would be the eye cells that would be the eye genes in a heart cell, or that would be the genes for ovaries in a man, right? Man doesn't have ovaries, so those genes are turned off. They're packed up and put away somewhere far away. And the reason how this is actually happened is due to these histones. So I'm not going to go into too much detail about histone acetylation and DNA methylation and all of that, but basically, based on chemical modifications to the histones, so the histones have these little tiny tails hanging off of them, and the DNA is wrapped around them. So this blue stuff is the DNA, and then these things are the histones. So the DNA gets wrapped around histones, and then all of these associate together, all these histones do, into something called a nucleosome, which is a bunch of histones and DNA in one structure. So the histone tails stick out of the nucleosome, and they can be modified. They can either basically be modified to be sticky to other histones or to be not sticky. If they are sticky, then they are unacetylated. They're sticky to each other, and then all the nucleosomes stick together, and it's really, really, really tightly packaged. If they are not sticky, the histone tails don't stick to each other, and the chromatin is more loosely packaged and much easier to get to. So this would be things for genes that we don't need to express them, and this would be genes that we are using in the cell's everyday functions. So the difference here is this is called heterochromatin, and this is euchromatin. This has the genes that we are going to want to express. This does not. This is packaged up in the attic. This is out on the coffee table. You don't need to know that. Don't worry about DNA methylation or histone acetylation. Okay, so then the next step, so we just packaged our DNA. So now we're going to turn that DNA into RNA. And so most of eukaryotic gene expression or control of gene expression comes from transcription. So it's mostly control of transcription. Which genes get made into RNA, they're basically going to get made into protein afterwards. So that's why this is such a big step. Oh, that's what I just said. Okay, so transcription is when we turn our DNA into RNA. And so if our RNA polymerase can't get to a gene because it's packaged so tightly in the histones, the nucleosomes are so close together, you put those books so far in the back of the closet you can't get to them, then the transcription initiation complex, the RNA polymerase, the transcription factors, cannot get to it. So they're not going to be transcribed. Okay, so basically we talked about transcription factors before. And what transcription factors are things that help bring the RNA polymerase to the mRNA. And so most eukaryotic genes are things called control elements, which are parts of the DNA which are non-coding, which means they're not going to get turned into a protein, that serve as binding sites for transcription factors. So that's where the transcription factor binds onto the DNA and then helps transcription happen. Okay, so there's different kinds of transcription factors. There's general transcription factors that will help RNA polymerase bind to any gene. This is like in a growing cell or in a growing embryo, we want a lot of transcription basically of anything that's available. We want a lot of transcription of all the proteins because we're trying to grow as fast as possible. So whereas if your cells are kind of stagnant, they're not dividing for whatever reason, then we don't want as much translation or transcription, so we don't want the general transcription factors on. So they don't lead to a ton of transcription of any one gene, the general transcription factors, but they will help turn on transcription overall. And then there are very specific transcription factors, which are gene-specific. So if I really, really want to turn on the gene for keratin in my skin cells, I have specific keratin transcription factors, which are going to bind to the enhancers, let's sit down a second, to DNA that, when they bind, bring RNA polymerase to the keratin gene. So the spaces where the transcription factors bind are called enhancers. Okay, this is more detail than we're going to get into. Basically, here's transcription factors over here. They bind to enhancers, which are kind of far away from the DNA, or from the gene that we're going to transcribe. Here's the promoter. So the activators are far away, but they bind over there, and what they do is then they help the DNA bend, which helps bring in all of the transcription factors and the RNA polymerase, and then RNA synthesis can happen. So basically what you should know from here is that proteins can affect transcription in lots of ways, even in areas that are not close to the DNA. So this section right here is super important for transcription of this gene, even though it's so far away from the gene. But all these proteins are necessary for transcription of this one gene to happen. So if we don't have those proteins, if they're not there, then this gene is not going to be transcribed. So that's how we can regulate it. Okay, so we don't have operons, but there are some things called coordinately controlled genes. So these are genes that are functionally similar. So maybe we need to break down a protein, like or maybe we need to build a protein or an amino acid, like the bacteria needed to build tryptophan. We don't have all those genes right next to each other, like the trip operon does, and they might even be on different chromosomes. But all of those genes will have special markers. They're called activators, I think. Basically, they all have these special markers which all bind to the same transcription factor. So if I say, okay, I really need to make some phenylalanine amino acid, all the genes needed for phenylalanine have a specific transcription factor. So if I send out that one transcription factor, then I'm going to make all the genes for phenylalanine. Even though they're not right next to each other, still a bunch of light switches, like in my kitchen, but I'm still sending out like one signal to turn on those light switches for the same thing. Okay, so after transcription is made, there's a lot of things that could happen, but transcription is the main regulator of gene expression. Basically, we have alternative mRNA splicing. We talked about this last section. Basically, we cut out certain introns in one gene, cut out certain introns in another gene, we get two different gene products. So for example, here's a gene for troponin. There's differential RNA splicing, so we could leave in exons 1, 3, and 5, and we get this protein. Or we can leave in exons 1, 2, 4, and 5, and we get a totally different protein that are do two totally different jobs. So whichever one we splice out, if we need this protein, then we're going to splice out 4. But if we need this one, then we're going to splice out 3. So that's another example of how to regulate what products we're making. And then how long we keep the mRNA around will dictate how much product is made. So if an mRNA is really short-lived, we're not going to get that much protein from it, right? Because it's not around for that long to be transcribed. But if we make sure that our mRNAs are around for a long time, then we're going to get a lot of gene product from it, a lot of protein. For example, hemoglobin's mRNAs stick around for weeks because we always need hemoglobin. It's what carries the oxygen in our blood. But there's a lot of genes inside of prokaryotic cells that are gone in a couple of seconds once a couple proteins are made because they don't need as much as we do. So if we keep our mRNA around for a long time, then we're going to get more protein from it. And if we don't, then we're not going to get as much protein from it. So that's another way to regulate how much protein is made. And that's based on the sections, the untranslated regions at the end of the mRNA. Also, we can regulate translation. So how quickly translation happens. If it's really easy to happen, then that mRNA is going to be made into protein right away. But if it's hard to put the translation initiation complex together, it's hard to get those ribosomes there, then we're obviously not going to make as much protein. And then after the protein's made, it'll be modified. So by adding a methyl group to one polypeptide, it might make it totally different than another polypeptide. So that's 2 different gene products. Or the protein might get broken down right away, so it's there to do its job for like 5 minutes, and then it gets degraded and it's not there anymore. Or there's some proteins that are never going to be degraded because we want them there all the time. So from DNA to protein, that first image from the cell, from this chapter, this section, shows all the different possibilities of regulation of eukaryotic gene expression.